Hey everyone, for those of you who are on the stream, we are a little bit delayed here, so I'm gonna add a couple more minutes to the timer, but Dr. Sutton is here and we'll be doing the presentation very shortly. So just wanted to give you guys an update.
All right, we are going to get ready to get started here with our first speaker of the day, who happens to be Dr. Sutton himself. Dr. Sutton is a professor of molecular and human genetics with Baylor College of Medicine, and he does see white Sutton patients. I just want you guys to know what an honor it is to have him here and that he is a part of our community. Oftentimes, when you have researchers or doctors who identify things, they don't always stay with them, and he has graciously come to our events, been part of our community, and has been a big help to us. So please give him a warm welcome.
Good morning, everyone. Thanks for the kind introduction. I'm really glad to be here with everybody today. And I certainly, again, want to kind of thank everybody that's helped to put this together. So Josh and Mandy, the Melissas, and Kevin, and everybody else for, again, making this a really great and lovely event. So special thanks to them. So for those that were in Orlando last year, some of this you will have seen already, but I know that there are some new people here this year, as well as hopefully new people online. So I'm going to kind of just now say first, like, welcome to Houston. So this is the Texas Medical Center. I'm kind of showing you where my office is at Texas Children's with the little green arrow there. And then that's downtown Houston in the back. So this is just about a mile or so down Holcomb right here from the hotel. So I'm going to kind of go through, particularly some people have asked me about kind of identification of white Sutton syndrome. And so I'm going to go through that just very briefly. So there were a number of studies, again, starting kind of about 2012, that were large cohorts of individuals with neurodevelopmental issues, including things like autism or intellectual disability, that suggested that that POGZ could cause various neurodevelopmental disabilities. These were big groups of individuals. There was not kind of individual level data on each individual. And again, there were only kind of maybe one or a couple of individuals in each of these studies. Similarly, there were variants, again, from a schizophrenia cohort as well. But again, like none of these were definitive. These were kind of what are called association studies. And so kind of where I got involved is one of my colleagues at Baylor, Jim Lupsky, works on a condition called Smith-Magenis syndrome. This is a condition that has, in addition to neurodevelopmental disabilities, a lot of sleep issues and behavior issues. And so, this individual that he had received a sample on did not have the typical chromosome deletion that you see. And so one of the graduate students with us, Jansen White, kind of one of his parts of his thesis project for his PhD was to try to look for genetic changes in this group of individuals who were thought to have had Smith-Magenis syndrome. And so there was this one individual who was a 15-year-old girl in the group, and she had this change in POGZ. There were samples on both parents, and neither parent had this change. And so Jansen then kind of came to me and, as the clinical geneticist for the research team, and we discussed it and said we should see if we can find other individuals. And so we asked our clinical diagnostic lab at Baylor Genetics, as well as another diagnostic lab, Ambry, if they had ever seen this. And so we found 4 other individuals with a change in POGZ who all shared similar kind of neurodevelopmental issues. I'm not going to go through this in detail, but you can see that there were various changes. Patricia talked about this yesterday. All of these asterisks that she had mentioned are changes that basically where the gene just kind of stops partway through when it's being made, and you can see that again. All of these were not inherited from a parent, and so those sorts of things supported that POGZ was causing the neurodevelopmental disabilities and other things that you see. Again, we talked about this. This is just again kind of from the original publication, and I'll talk in more detail about these issues. One of the things that's a pretty unusual birth defect is something called diaphragmatic hernia. So that's a hole in the diaphragm. And that, again, has been seen repeatedly. It's not common, but it is an unusual birth defect and has been seen in more than one individual. And then shortly after we described those 5 cases, there was a group from Europe that had collected a larger cohort of 25 individuals. And so basically, in looking at those kind of 2 original groups, our 5, and then subsequently their 25, this is just a summary of what you see there, that there is a range of intellectual disability that is seen. About half have features of autism. You again heard from Astrid yesterday about these other challenges, particularly obesity. I'd mentioned the sleep disturbance. Some had changes on brain imaging or brain MRI, and then I'll talk about sleep on the next slide. So one of our former trainees shown here, Reid who trained with us and is now back home in Israel. When she was a trainee, worked with me to put together a survey that the foundation graciously sent out to try to get more information about various issues, particularly developmental issues, sleep, and then GI issues that we had heard a lot from patients and families of being a concern. Again, some of this looks similar to what you saw from Astrid yesterday from the Simons Foundation registry. This is showing kind of on the on the y-axis. Okay, good. So on the what's called the y-axis here, this is age in months, so 0 up to 70 months. And you see for various assessments of things like gross motor skills, you see that that again, there is quite a wide range for some of these things, particularly language development, where some are only kind of mildly delayed to first words, but then others have more significant delays until achieving some of these language skills. Again, we had heard a lot, so we actually had a specific sleep and GI or stomach concerns questionnaires that were standardized that people were kind enough to complete. And so you can see constipation in about half, but as well as feeding difficulties in about 2/3, again, close to half having cyclic vomiting and other sorts of concerns that we know about. And then on the pediatric sleep questionnaire, about 1/3 had sleep apnea as well as other sleep disturbances. This is, again, another publication from now just a few years ago. This is a group from France. And again, this is kind of just going through the various developmental issues, and again, kind of the wide range of developmental issues that's seen, again, kind of in more individuals. A lot of people kind of ask me about genetic variants, and so I'm going to spend just a moment or two talking about that. So we all have variants in our DNA, and that's what makes us unique. I say sometimes it's kind of like a meatloaf recipe. Everybody makes it a little bit differently often, but it can still be delicious, made multiple different ways. So, our genetic information is like that there are changes that we can have in our genes that make us unique in various ways, but some of which changes can cause challenges in a unique way. And so in regards to kind of when you're looking at a genetic report, the laboratory should follow standard guidelines in how they define a variant. And so this is kind of a list of the terms. So if you see pathogenic, that means that change is known to cause White-Sutton syndrome or whatever condition is being tested for. If it says likely pathogenic, that means there's very strong evidence that this, again, is indeed causing White-Sutton or a particular condition. But then there are these other results called one variant of uncertain significance, meaning there's just not enough data to know if this is disease causing or not. And then likely benign means it's probably not of any interest, and benign means it's definitely not of any interest. Most all of the changes, again, you saw this from Astrid yesterday, are stop variants. And so when you're, again, looking at a report, you will either see kind of an asterisk, or if it's an older report, maybe an X or a TER. And so again, this means that the part of the way through of making the POG-Z, that it just kind of stops, that it kind of, part of it is made and then it stops partway through so that it's not made. And in most of these cases, the body is going to recognize, oh, this isn't right. It's not the whole thing. And so it'll actually be degraded or broken down. If you see de novo, that means that they've tested parents and that it's not inherited from the parents. People a lot of times say, well, I thought everything was inherited. What happened? So what occurs when people make kind of egg or sperm cells to pass on their genetic information to their children is that all of the genes that they have need to be copied. And so this is kind of and if it's de novo, this is when the genes are being copied, there's an error that's made in that copying process. And so we have 20,000 genes. You can imagine that it's probably a wonder that more mistakes aren't made if you're copying kind of 20,000 different genes. But again, this is kind of how it can happen and not be inherited from a parent is, again, it's just a mistake that's made in copying that gene. This again is just showing Astrid, had a similar image yesterday, but again, the variants occur. This is the various sections of the gene shown here in these boxes. And then you can see that the various changes kind of occur all along the gene, but do tend to cluster more toward the end of the gene here. And this is showing various regions. Again, this gene is important, and it basically kind of regulates how other genes are expressed. And so all of our genes work together, but this gene, particularly POGZ, is working to kind of help other genes work or be expressed. Again, sometimes, particularly if there are these variants of uncertain significance, we want to kind of try to resolve that. And so I'm going to kind of show two different ways potentially to resolve that. So this is a patient that was enrolled in our local undiagnosed diseases network. She was found to have a change that's kind of unusual. So you don't see like that asterisk or X or stop sort of thing here, that this is actually a change that's not even in the coding part of the gene. It's in the part of the gene that kind of puts everything together. But it's so this was kind of an unusual change, and so we weren't certain whether this was of interest or not. But again, what Lindsay, along with a visiting medical student for the summer, Ashanta Merriweather from Morehouse, did is to do something called RNA-seq. And so this is, again, kind of sequencing the messenger from the gene. The green and the blue shown here are normal. So you can see that these big boxes, again, are the coding parts, and these arcs are showing how they're linked together. And the green and the blue are normal, where you have this arc here and this single arc here. But now in this young girl, you can see that there's this other arc here where you're skipping this part of the coding gene. And so again, and that happened close to half the time. And so again, this supports that, again, this change in the non-coding region is indeed disease causing. The next thing, so this is actually a slide that's courtesy of a good friend and one of our former trainees, Bekim Sadikovic, who's in Canada. So one of the things that he works on is looking at how changes across genes are seen in something called methylation or epigenetics. And again, this is kind of how our genes are regulated. So we don't just, all genes are not turned on all the time at the same time and in all the same cells. So there are some genes that are, let's say, turned on in nerve cells but are not turned on in liver cells. Or there are some genes that are turned on, let's say, early in development that are turned off by the time we're born. And so these changes in DNA are what kind of make sure that the genes are working in the right place at the right time. And so he's defined this sort of pattern for many different diseases, a lot of which, again, are very much like POGZ, where, again, they're genes that are regulating how other genes work or when they work or how they work. And so he's identified a series of profiles that is diagnostic for a wide variety of conditions. So this is, again, not published yet, but again, he was kind enough to let me share it today. And so what you see, we'll kind of focus mainly on this area here. I think this is probably easiest for a non-scientist and even for sometimes a scientist to understand. So the red dots are, again, these variants in POGZ that are kind of known to be or are very likely to be disease-causing. This kind of orangey yellow are these variants of uncertain significance. The purple ones are, again, kind of confirmed known variants, what are they called, a validation sample. And then these bluish green ones are non-white Sutton or non-POGZ variants. And so what you can see here is in these kind of two different coordinates using this, you can see that a variety of these are all kind of clustering together here. And you see that there is this clustering that's distinct from the control samples. And you also see that, again, there are some of the variants of uncertain significance that are clustering with the known patients, indicating that they are likely disease-causing. And then there are other of these orangish variants of uncertain significance that are clustering with the control samples that mean that these are likely benign or likely not disease-causing. This is a test that is, the name of the test is called EpiSign. It is clinically available. And so if there are individuals that have these variants of uncertain significance, this testing is clinically available to try to help resolve that, to know, again, if it is disease-causing or not. Frequently people ask how common it is. So again, another one of Reid's projects when she was training with us was to go through over 12,000 patient clinical samples that we had seen in our diagnostic lab for genetic testing called exome sequencing, which is sequencing of the coding regions of all 20,000 genes, to see how often there were changes identified in POGZ. And so this is, again, just kind of showing the category. Most of the reasons for testing when changes were found were, again, these neurodevelopmental changes that are shown here. And so kind of in the interest of time, I'll just kind of summarize that of individuals that were tested in our lab for a diagnosis of either kind of autism, developmental delay, or intellectual disability, only about 1 in 500 actually had a change in POGZ. So this, again, is as I think everybody knows, is a rare condition. So now I'm going to kind of quickly move through a few management issues and kind of recommendations. So again, for the developmental delays, early engagement with therapy services. Many states have this for free for the first few years of life, and then through school systems, even kind of in the kind of pre-K age range. If there are concerns about particularly things like walking or motor skills or mobility, there are specialists, either kind of pediatric orthopedic surgeons or a specialty called physical medicine and rehab that can help with various adaptive devices. And I know on the video last evening, we saw a number of individuals who had those sorts of things to try to, again, help them with their development or with mobility. Again, kind of early intervention and making sure that that the school has education plans such as a 504 plan or other sorts of things. And again, working with local doctors to kind of work with the school as needed to help with that for individualized education needs. If there are particular challenges with autistic features, again, there's a program called Applied Behavioral Analysis, or ABA, that can help individuals with socialization and behavior when they have those sorts of autistic behaviors. Again, there are a variety of medications that can be used. I typically would start with one called Risperdal, but there are a variety of other medications. One of the things that often is challenging is that most all of these medicines that are used for behavioral issues are metabolized by our liver. And within the population, there is a wide range in how people metabolize things. So, you maybe sometimes say like, oh, you hear people say like, I take Tylenol. I tried taking Tylenol for my headache. Tylenol never works for me, right? We all metabolize medications very differently. And so there are some individuals that maybe are what are called fast metabolizers that kind of get rid of the medication very quickly, that need a much higher dose. And there are other individuals maybe that are what are called slow metabolizers, which would mean that basically even at a low dose, they could get either kind of side effects or and so kind of working with a psychiatrist or other professional to kind of try different medications and making sure you're keeping in contact with them about behaviors, and so that they can either kind of change the dose or change the medication. In surveys, there are not specific medications that have been identified that work for everybody that has seizures, but again, kind of working with the neurologist to again kind of manage medications for seizures. Hearing and vision, as you've seen, can be concerns for a number of individuals. So again, making sure that those things are monitored by ophthalmologist or audiologist for hearing to, again, ensure success at school, that they can really be fully engaged. Because again, those things can certainly kind of exacerbate behavior issues if they're having challenges, hearing at school or hearing at home, as an example. Sleep disturbance, again, is common with sleep apnea seen in a third. So one of the things that's important, if you particularly notice a lot of snoring at night, kind of waking up in the night, stopping breathing, pauses in breathing overnight, a sleep study can be performed. This is typically done in a kind of a hospital or clinic setting where overnight they monitor kind of brain waves and breathing and all sorts of things to make an assessment, and then can recommend interventions. One of the things that certainly can be tried over the counter that works for some people is melatonin, which is something our body makes normally as a hormone to help with sleep. And so melatonin at bedtime can help, again, if there is obstructive sleep apnea, to, again, work with a sleep specialist to manage that. And then there's a condition called restless leg syndrome that can often be caused by low iron levels. And so, again, if that's noted, again, on a sleep study, that would be indicated to check blood iron or something else called ferritin, which is an iron-related protein. And then gastrointestinal concerns, again, we saw are quite common. So again, working with the gastroenterologist to manage things like constipation or other things. Some individuals have reported that, again, kind of treating with Tylenol for the cyclic vomiting can be helpful, and it's thought that this is maybe because this is something called an abdominal migraine. Also, again, particularly if vomiting is a major concern, there are medications that can help with nausea and vomiting. Kind of the more common one is called ondansetron. This is very safe. It comes in a little tablet that dissolves under the tongue. And so, again, if you're having a lot of challenges with cyclic vomiting, maybe kind of having this at home for when it happens can help avert an attack, or at least help avert the need to kind of go to the emergency room or to the hospital. Again, kind of regular management of constipation is important. So if constipation is a challenge, kind of having a regimen that is done every day. Sometimes people will kind of say like, oh, well, things are going pretty good, and stop. And then the constipation will come back. And so kind of if constipation is an issue, just again, kind of regular monitoring. And then feeding therapy. And again, if feeding is an issue, or again, particularly cyclic vomiting, sometimes what's called a gastrostomy tube or a G-button can help families to, again, be able to manage these things at home and have less frequent visits to the emergency room. And then finally, again, obviously the importance of community and patient advocacy groups, but also other things like Special Olympics to, again, kind of get kids involved. And then, again, kind of resources. Obviously, there are great resources on the White Sutton Foundation website. I'd mentioned to some people, again, there are other resources from, these are both from the National Library of Medicine. The first one is really kind of more intended for healthcare providers. And then this second one, Medline Plus, is really more for patients or families. But there's a lot of good information on both of these that could also be shared with like teachers or other individuals. And then there's also information on the National Organization of Rare Disorders, or NORD, with the link here. And then finally, for research, again, there is a federal website called clinicaltrials.gov, where all research that's either kind of funded by the federal government or that's going through the U.S. Food and Drug Administration is required to be posted, but many people kind of post these electively because they know it's a resource that many people use to try to find research studies. And so again, I want to recognize everybody that helped us along this journey, particularly Jansen White, shown here, my colleagues Jim Lupsky and Jen Posey, and then you'll hear from, I think, Paolo and or Anna later today. So thanks. I'm happy to take any questions if there's time.
Thank you, Dr. Sutton, so much for that.
Have y'all found two people with the same genetic?
That's a great question. So I'm not make sure that everybody online can hear. Melissa was asking if there are two people with the same change. There are some. There's not a change that we see with high frequency. So there are certain conditions that we know of where there are a relatively small number of changes that everybody has. So kind of probably the best example people may have heard of is sickle cell disease. So there essentially is kind of two changes, and everybody with sickle cell disease has one of those two changes. But there are other disorders, again, like White Sutton, where there are kind of changes all over the gene, and that there's not kind of one change that's like really common. But there are a small number of individuals that do share the same change.
Does it persist?
Yeah, I think it's not. So it's a challenge because there aren't enough individuals with that same change to really know. They're certainly not identical,
Right.
but I think it's a challenge to know, like, is this change really different from all the other changes that we see in some way? Because again, it's so infrequent for people to have the same change.
Once again, thank you for all you've done for us and helping us. Yesterday was kind of unique when we were looking at the Simon's charts and seeing how small the numbers are, and seeing how it's finally getting across the world. You're starting to see more things. What's the best way to get out to the rest of the world, get those numbers up so we get a better class to look at? And what can we do as far as, other than the blood draws, the testing, and, we have our community support amongst us, but how do we get into the government, the CDC, different groups to really push this to another level for funding sources?
Yeah, so I think, again, really kind of, using all sorts of tools like social media and other things to really kind of encourage people to participate. The Simons Foundation, again, is a great partner for that. And again, I don't know that Astrid kind of really talked a lot, but they work with a lot of different organizations. But again, kind of they are generally quite well funded from the Simons family. And so that's a good resource. The National Organization for Rare Disorders, or NORD, that I'd mentioned earlier, they also will have kind of some support for like registries or databases that they could do that. And then again, I think for like research grants, again, kind of working with researchers to really try to support them. And again, part of that is always going to be trying to form a partnership. So researchers need samples from things like the Simons Foundation or other things, right, to conduct their research or to understand. And so again, that sort of partnership can be very helpful. For example, with a research grant, right? That if when people write research grants, we often kind of ask for letters for collaborators, right? And so this can sometimes be very helpful if the people reviewing the grant see, like there's a commitment from, the Simons Foundation to provide 30, lymphoblastoid cell lines or whatever it is that the researcher would use. That those sorts of things are generally viewed very favorably in review. There are, of course, other things that they look at, but again, kind of partnering with researchers and again, to make sure that they have the resources, the patient resources like blood samples or other things that they need to be successful.
All right, this will be our last question.
so we can move on to our next speaker.
So I have a question as far as that variant of insignificance, because myself and my daughter have that. How long you said there's more testing that we may have to do, or?
So one of the things that I meant, and one of the reasons I wanted to mention this EpiSign, and again, I want to thank Beckham Sadikovic for providing that, is so that's a test that, again, is clinically available that could be done to try to help resolve that. So when the lab classifies those variants in those different ways, including a variant of uncertain significance, there are standard protocols that they follow. But again, something like the results of like an EpiSign test could help the lab to be able to change that. So now it's not a variant of uncertain significance, that it's, either kind of pathogenic or likely pathogenic or benign or likely benign.